Transcription is the first step in gene expression in which the formation of a gene is used to construct a functional product such as a protein. So the main enzyme involved in transcription is RNA polymerase, which uses a single-stranded DNA template to synthesize a complementary strand of RNA. Specifically, RNA polymerase builds an RNA strand in the 5' to 3' direction. So initiation, the polymerase binds to the sequence of DNA called a promoter, and then once bound, the polymerase separates the DNA strands, providing a single-strand template needed for transcription. Elongation acts as a template for RNA polymerase as it reads the templates from one base at a time. The polymerase builds an RNA molecule out of complementary nucleotides, making a chain that grows from the 5' to 3' end. Termination, the sequence called terminators signal the RNA transcripts that it's complete. Once they are transcribed, they cause the transcripts to be released from the RNA polymerase. In bacteria, RNA transcripts can act as a messenger RNAs right away. In eukaryotes, the transcript of a protein codon gene is called pre-mRNA and must go through the extra processing before it can direct translation. Eukaryotic pre-mRNAs must have their ends modified by adding 5 caps at the beginning and a 3' poly A tail at the end. Many eukaryotic pre-mRNAs undergo splicing. In this process, parts of the pre-mRNA called introns are chopped out, and the remaining pieces called exons are stuck back together. End modifications increase the stability of mRNA, while splicing gives the mRNA its correct sequence. If the introns are not removed, they'll be translated along with exons, producing a gibberish polypeptide.
A genetic material is called DNA. If we unwind the DNA, we see that it is a double strand made up of nucleotides. There are 4 kinds of nucleotides that make up the DNA: adenine, thymine, cytosine, and guanine. We replicate DNA during mitosis. Our DNA contains many genes. The genes make proteins and enzymes. To do this, the body needs to make RNA. Our body helps make RNA with the help of an enzyme called RNA polymerase. RNA polymerase synthesizes RNA by using one of the DNA strands as a template. This process is called transcription. RNA has a poly A tail, which means it has many adenosine nucleotide and at one end. On the other end is a 5 cap. The particular RNA that was synthesized is called mRNA. It's a pre-RNA. There are 3 major types of RNA in humans: mRNA, tRNA, rRNA, and rRNA. They all synthesized by different RNA polymerase. They also contain mRNA then goes through splicing. Splicing is when the spaces of introns are removed in mRNA. The exons will split. This will form a new kind of mRNA, therefore a protein. mRNA will then leave the nucleus through a nucleus port into the cells of the cytoplasm. mRNA serves as a template to form proteins. mRNA is like a cookbook which is used to create proteins. Now we pause on mRNA and go look at another. The RNAs are important because they are in the process of protein synthesis. A specific part of DNA, which is created with RNA polymerase III, is synthesized to transfer RNA in the process called transcription. It means getting rid of the introns and leaves a type of tRNA. tRNA then leaves the nucleus through a nucleus port. The tRNA will fold into a kind of T shape. Since the tRNAs are made of nucleotides, and what every tRNA does is attach to one amino acid which are in the cytoplasm. If the codon and anticodon match, they will bind. The ribosome keeps moving, and the tRNA will then ride on the P site of the ribosome. The ribosome lets go of the amino acid from the tRNA in the P site. The ribosome keeps moving, and the tRNA will exit the P site. The amino acid that was removed joins the other amino acids from the tRNAs to to form a polypeptide, aka a protein. These can be broken down to amino acids so the cycle can continue. The ribosomes are translating the whole mRNA to make protein.